Okay, hello and welcome everyone to the varvis webinar series. Good morning, good afternoon, wherever you are. So today's session is about the challenge of variant interpretation. We have three experts here joining us for the webinar, and they will tell you from their own perspective what is necessary to accept this challenge successfully in clinical routine. The webinar is divided in three parts. The first part will be more the clinical perspective, variant classification, ACMG criteria, and finding the right evidence, presented by Dr. Andreas from the NGS in Munich. The second part is on how to master the flood of new data, presented by Dr. Ben Liesveld from Limbus Medical Technologies. And the third part is on comprehensive literature search, the entire world of genomic evidence at your fingertips, presented by Dr. Markier from Genomic. We will conclude with the Q&A session, and we encourage you to participate in the discussion and ask all the questions you have in mind. To do so, depending on the device you're using, you can enter your questions from the mobile device behind this question mark icon in the header. If you're using a desktop version, the question mark is in the right panel next to the presentation. Additionally, we have more information uploaded for you. So behind this sheet icon, there are handouts that you can download for more information. Before we get started now, let me briefly introduce to you who we are. As we are hosting this session today, my name is Yvonne Kassmann. I'm product manager here at Limbus. We are located in the north of Germany, right at the Baltic Sea, as you can see here on this map of Europe on the right. We are a software development company and a medical device manufacturer with a young team. We develop and operate our software according to international medical device standards. Our product varvis is a cloud-based clinical decision support system specifically designed to aid in the diagnosis of patients. varvis is a complete software solution for all kinds of NGS data. It is a cloud-based medical device software, which means that there's no investment necessary in the IT infrastructure, so we can get you started within a couple of days. We provide equal performance and support across the globe, and also immediate QC feedback to optimize your laboratory processes. varvis scales well from small panels up to whole exomes and also whole genomes. And one of our key features is our validated and automated CNV analysis, which allows you to replace conventional CNV detection methods in clinical routine, and also this offers a lot of cost savings and time savings. Our customers are laboratory service providers and university hospitals across the globe, mostly located in Germany, and one of them is the NGS, the Medical Genetics Center, which is an owner-managed practice and laboratory for human genetics with headquarters in Munich. They were founded in 2000. They have more than 130 employees at the moment, and they report. They provide reports to more than 10,000 cases per year. At least these were the numbers from 2019. Within the team, there's a 60-member medical team offering genetic consultations in Munich and 9 other locations in Bavaria. It's an affiliated, accredited laboratory, and the examination covers the entire spectrum of genetic diagnostics, as you see below. It's a very well-known and lab here in Germany and also in Europe, and it stands for high quality patient care and consultations in human genetics. And from the NGS, we have here today with us our first speaker, Dr. Andreas. I'm happy to introduce to you. He did his PhD in molecular genetics and has more than 20 years of experience in the fields of cancer genetics and variant interpretation. He joined the MGZ quite early on, I think 16 years if I remember correctly. And since 2014, he's the head of the genomics program there. Besides, he has many other engagements. For example, he's the co-chair of the Insight Variant Interpretation Committee and a member of the organizational committee for the HGBS Variant Effect Prediction training courses, which are held across the globe. He's also a member of the scientific program committee for the ACMG symposia and an active submitter and curator of the LOVD database. So today, he will give his own perspective on variant classification, ACMG criteria, and finding the right evidence. Thank you, Andreas, for joining us, and I'm handing over to you. You are mute, I'm sorry. I cannot hear you. Sorry.
Does it work right now?
Yeah, now I can hear you. Thank you, yeah.
Okay. So maybe therefore we made the test, but now it's not working, so we make it free. So thank you for the kind introduction. And yeah, before I start, do I have the control over your slides?
Yes, I think so.
I don't see anything right now. It's still okay. So before I start, I have to make you understand that I'm not a member of the ClinChen consortium or the SVI working group. But as already mentioned, I am a member of the Insight Variant Curation Expert Panel. And as such, I was involved in formulating the Insight classification rules, which deals with the polyposis genes and all colorectal cancer genes like the Lynch syndrome genes. And you were actually one of the first ClinVar approved 3-star expert panels. And yeah, such modifications of gene or disease-specific ACMG codes is indeed a very important topic, and I will come to this later in my presentation. I want to give you a short introduction about. I try to find the pointer. There we are. I have a very small screen. Okay, so I will start with a short introduction about variant classification in general. Then I will jump into the ACMG-AMP classification system, and from now on I will only talk about the ACMGs and skip the AMP parts to save time. Then I will discuss some of the pitfalls and challenges by using these ACMG rules. And at the last part, I will show you how we use Mastermind and varvis together for gathering ACMG relevant information. And maybe some of you know that the idea of grouping variants in a 5-class system originally comes from the IAC group. So the IAC stands for International Agency for the Research on Cancer. And they proposed 2008 in this paper from Sharon Blorn et al. To classify variants in 1 of 5 classes. And more importantly, all of these 5 classes are linked to validated measures of causality. And these are expressed as the probability of being pathogenic. More importantly, all of these 5 classes are linked to clinical recommendations, whether to test relatives, how to do recommendations for surveillance, or if you should test family members at risk in a research setting. So the ultimate goal of IAC was to give actionable clinical recommendations to genetic data. And this was not only meant for cancer genes, but they proposed that these 5-class classification system should be used for all other genes as well, for all diseases. And they conclude that an accurate and consistent variant interpretation and classification is a prerequisite for genetic diagnostics and also precision medicine. I will now come to my first personal and most important rule in variant interpretation. Say you detect this variant in the BRCA1 gene, and you get from a narrow literature search or from consulting ClinVar or LOVD or your colleague, the opinion that this is a class 5. Well, it actually is a class 5, but I want to make this point by showing you first the first rule of Fight Club. Maybe you will remember this iconic film from the late 90s with the young Brad Pitt. So the first rule of Fight Club was, we don't talk about Fight Club. And in variant interpretation, it's actually exactly the other way around. So my first rule in variant interpretation is you never ever uncritically adopt the classification of a variant from a database, the literature, your colleague, any other smart tool which is now available and around, but you always try to identify the underlying evidence, and you definitely talk about variant interpretation. This can be exemplified by showing you that we can reach this class 5 by very different pathways. For example, you can use the Enigma, VSEP, or Variant Curation Expert Panel classification, Their calculated multifactorial likelihood of 99% translates to a class 5. Alternatively, you can use the standard ACMG-AMP rules. So Enigma used the ACMG codes and modified it to the gene-specific requirements, but you can also use the standard ACMG codes, and then you come to a class 5, or you can even use your own laboratory classification system. So maybe you noticed that some of the big US labs, they develop their own classification rules, but as long as they are transparent, published, and you can see why they choose some criteria, you can follow them, and that's valid. You can do this. If you submit variants in databases or on the report, I personally vote for displaying the ACMG codes you invoked, because so in this case it's easy. It's a clear-cut class 5, but if you have a VUS and you have a functional study in your lab, or you have a large family with positive co-segregation, you could deliver the missing piece or the missing stone here and clear the case. So what are the ACMG-AMP rules? They provide a qualitative evaluation of different data types. We have 28 defined criteria, and each one has an assigned code. Each code then is assigned a weight, even a standalone, very strong, strong, moderate, and a direction, benign or pathogenic. And the variants can then be assigned to one of these 5 classes I showed you before. And if not enough lines of evidence are invoked to classify a variant, or if there are valid but contradictory lines of evidence, a variant is interpreted as a VUS. This last point often points to low penetrance or reduced penetrance variants, but I will come to this topic later. So this is what it looks like. The ACMG-AMP rules come with a table, and you have here the different data points where you can look for evidence. For example, functional data, segregation data, or population data. And here from far left to far right, you have the evidence codes with the weight, either being strong benign or very strong pathogenic. And there's also a table where you combine the evidence codes you invoked. So for example, let's say you have 2 moderate criteria and 1 strong, then this would combine to a class 4, likely pathogenic. And this is an example just to show you one class 1 in TP53 and in SPAST1 gene a class 4. And it's not that rare that at the very last point, the molecular biologist makes all the variant interpretation properly, but in the very last point of calculating the evidence codes together, especially if you adjust one evidence code, maybe come one slide back, I forgot to mention that almost every one of these specific codes can be adjusted in strength according to expert knowledge or published rules. So if you adjust, for example, a supporting evidence to a moderate and a strong one, you have to keep that in mind, the last step to calculate it. So what I like in the varvis software tool is that here's a screenshot from the variant classification window that you can adjust the codes you found, and you can then adjust the weight, even very strong, moderate, supportive, whatever. And and if you don't use a tool like this, then you can consult, for example, the ClinGen homepage. There's also a pathogenicity calculator tool there. So before we go deeper in the ACMGs, I have to give you some general considerations or disclaimers. I want to say them, because these are often the sources of error when you work with the roots. So the general considerations is that these roots are intended for variants in all Mendelian genes, whether they are detected in a single gene assay by Sanger, gene panel, exome, genome, or even transcriptome. But very important, the genes must have penetrant, they must be penetrant enough to be recognized, and also they have to have a recognizable phenotype. Otherwise, you can get problems by applying the codes. And you must be very careful by applying these rules to candidate genes, so-called genes of uncertain significance. So as of June 2020, there were only 4,285 genes associated with a disease of phenotype. Also, the term mutations and polymorphism should not be used. Instead, the term variant with one of these qualifiers should be used. And of course, all variants should be reported using the HGVS nomenclature. In conclusion, the pathogenicity should be determined by the entire body of evidence in aggregate, including all cases, not only the one in front of you, but also the one in the literature or in databases, and you should arrive at a single conclusion. So what do you need to get the information you need for the classification? Well, obviously, literature is the most important area where to look for evidence, but unfortunately also the most time-consuming one. And this is the reason why we speak here today, or why I present this, because I will show you how you could use Mastermind to have sort of shortcuts to the relevant points of information. You can also use, of course, PubMed or these free available tools, but in any case, it's time-consuming. Of course, population databases are also very variable source, and I cite here Genomic, but Genomic is not only a population database, it's so much more, but this would be an entire other talk, so I will skip this. Of course, local-specific databases or disease-specific databases are also very important here, and if the evidence is provided there, you can find, like in the literature, almost all evidence codes. Last but not least, of course, the predictors come in, but they are not that high valued here. So speaking of variant curation or the ClinChen consortium in general, I cannot go to the time limitations in great details into in all recommendations of the sequence variant interpretation group, but you have here the link to the clinical genome homepage, the ClinChen homepage, and when you navigate here to the working groups, and drop down to the sequence variant interpretation working group, you find all recommendations regarding the modified ACMG codes. And also further down, you find publications. Some of these are summed up in publications. The SVI is led by Les Biesecker and Stephen Harrison, and many of the examples I show you today are from Stephen Harrison's slides. He has also provided very good tutorials in YouTube, and you can find them by navigating YouTube to the ClinChen tutorials. So as I mentioned, the ACMG-AMP codes in the beginning were rather broad, and it was well recognized by the authors of the ACMG-AMP rules that over time these broad rules had to be specified by gene or expert-specific working groups. So this is just a short screenshot. So the list is very long, but these are only 2 screenshots from the clinical domain working groups, where you can find in the cardiovascular setting or in the cancer setting or wherever, and you can find all the specific disease-specific expert panels and working groups dealing with the refinements of the ACMG codes. And this is important because here in this slide, I show you the contribution to discrepancies. So early after 2015, when the ACMG paper was published, there were some studies comparing different labs using the ACMG codes, and they found large discrepancies. And here are the groups where most of the discrepancies come from. And you can see from the wording that this is error prone. So for example, here it says minor allele frequency is too high, or here it says well-established study. So who is to say what is too high? That is only a question who can be answered by a gene-specific expert panel in my eyes. The well is true holds true for well-established studies. So this is a point where the experts should further refine the criteria here. And this has been suggested. So here in this screenshot, you see in green, surrounded with green, the criteria being modified by the SVI itself. In red, the criteria which have been removed by the SVI. So these were essentially the 2 reputable source codes, and this is exactly in line with my first rule in variant interpretation. So there is no reputable source. You have to look for the evidence, but you don't trust anyone. And in purple are the criteria which require gene-specific or disease-specific specification. The slides I showed you just before. And now I want to come to the problem, or well, I should not say problem. We at NGS, we don't have problems. We only have issues or challenges. So we have here the issue of subjectivity, and I want to explain what I mean here by this example from ClinVar, this variant in the CHEK2 gene. And you see here we have 14 submissions. 3 of them, they say it's uncertain and no, likely pathogenic, and 9 say it's uncertain. And to demonstrate what I want to tell you here is this is a screenshot, or this is an extract from one of the big US labs. I don't remember, maybe it was GeneX or Embry or Invite, and they provide sometimes very good sum ups or interpretations about the variant. But what I want to extract here is I did a sort of color coding, and I want to point here, for example, it says that this variant has been reported in individuals with a personal or family history of breast, prostate, pancreatic cancer, and so on. So these cancers are well in the spectrum of pathogenic CHEK2 variants. And you have also some literature citations here, a lot of papers. But what is it now translated in ACMG? Is this could we use PS4 here, overrepresented in diseased versus in controls? Should we reduce the strength to moderate or supporting? That's you to say. In green, it deals with the functional analysis. They have shown that indeed this variant leads to a reduced phosphorylation and kinase activity, as well as loss of DNA damage response with 2 papers. So is this strong evidence? Is this a well-established functional assay or not? Should we reduce it to moderate or supporting? Here in the red part, it says that the frequency is, it's not so rare, it's 0.3%. And these are exact data. I checked the variant yesterday, and now it's almost it's a couple of times homozygous in GNOMED. So cancer is not rare. This variant is not rare. Is this variant rare enough? Is it PM2, or is it too common? Or does the homozygous finds in GNOMED justify to evoke BS2? In the blue part, it says that this variant is located in a SQ-TQ domain. Is this a domain without benign variation, which is functionally important? Is this a very good defined domain? So can we rise the evidence to strong or reduce it to supporting? So this shows that there's a big issue of subjectivity. So we can look at the same data points and come to different conclusions. And I choose this example, but this is a rather hard example because it's CHEK2 is a known gene for strongly reduced penetrance for cancer. So as I mentioned earlier, so these reduced penetrance genes are sometimes very hard to classify, even if you have such a good sum up here. The next issue I want to discuss is the issue of rareness. This is a screenshot from all ClinVar submissions. And you can see here that 64% of all ClinVar submissions are unique records. So only one submission, so only one variant here. And here is some data from the exact data set. Unfortunately, I don't have the numbers from GNOMED, but from the exact data set, 99% of the variants have a frequency of less than 1%. And more than half percent of the variants are only seen once in the whole data set. So this shows you that most of the variants we find in a diagnostic setting are incredibly rare, and there's a large chance that you never see this variant again. And this is a really challenge in variant interpretation. So now I come to the more rather philosophic part of variant interpretation, and I want to raise the opinion or the statement that we should be aware that we are dealing in molecular diagnostics in an environment with incomplete information. So we learned a lot over the last 20 years or so, where I follow the field, but still we don't know enough, and we should have a more humble attitude here and admit that we don't know exactly the details. So I want to make this example here by showing you that we have to still have the problem that positive correlations are much more likely to be reported than negative ones. There are some initiatives right now in the Global Alliance and also in the HGVS that this should be changed. but still this holds true for all publications right now, and this can lead to an ascertainment bias amongst others. Also, we have to deal with the Rareness curse, which says that initial studies, this has been a problem in the cancer field for a long time. So initial studies using patients cohorts, which are sampled under the ascertainment bias, they're often enriched for high prevalence variants and high relevant phenotypes. But these initial studies show excellent clinical sensitivity. But if you open the box and look in larger control databases, then you see that this clinical sensitivity drops dramatically. So therefore, the evaluation process should include a very critical evaluation of all the published studies, and you should keep these two issues in mind here. So this you don't have to read this book. So this book was a present from my supervisor for my PhD thesis. I think he gave this to every one of his students. And so the core message of Karl Popper is that we as the scientists, and we are looking for ways to test the theories against the observational evidence, whereby test means attempt to refute the theory and not to prove it. So sometimes in our business, we want to solve the case. We want to give the physician a positive report. We want to find something. And this is not only a philosophical question, but I want to show you in the next slide that it is relevant for your daily work in variant interpretation. So this is an example from my lab. I had this case a couple of years ago, and I found this BRCA1 variant in this case, in this patient. It was a young female with breast cancer, bilateral breast cancer, and a highly suspicious family history. The mother and grandmother died, and the sister was also affected. And yeah, I found this variant. And for those of you who work in the cancer field, the ring domain rings a bell, because in BRCA1, there are only two domains where missense variants are pathogenic, and this is the ring domain and the BRCT repeats. So my first idea, and my idea which I preferred most, was that this variant is pathogenic indeed. So I looked for evidence for being pathogenic. Of course, this variant is very low allele count in GNOMED, so frequency is very low. And one missense variant, immediately 3' is described multiple times as being pathogenic. The predictors, they all say it's pathogenic, and I have a positive segregation. I found it also in the sister, which has also breast cancer. So if you remember Popper, if my favorite idea is that this variant is pathogenic, then I should do everything to prove that the opposite. And is there evidence for being benign? Well, actually there is. So a variant being rare is good, but it's not enough. So each individual has a lot of variants. And the missense variant immediately 5' is a benign variant with also a higher allele frequency. The prediction, well, it's only a prediction. It's only supporting evidence. And the chance of a co-segregation is only 50-50. But moreover, this person, so she carried also a truncating clear-cut class 5 in part B2. I don't say that this variant is benign. It could be that this woman carries two pathogenic variants. But if I would not find this part B2 variant, I would have been maybe more inclined to report this as a pathogenic one. So this is what I mean with don't try to solve the case. Okay, now I come to my last part, and I think I'm a little bit short in time, so I will try to hurry up a little bit. So this is a screenshot from varvis, and to make the point, I want to just show you what I did when I found this clear pathogenic variant in the ABCA4 gene. And so hurry up. So the phenotype fits very well to this variant, and I was quite sure that this is my first pathogenic variant, and I was looking for another one. I got two other variants in this gene. So this one was not very sexy, I would say, -25, but this one is a missense, is also a high score. And I was wondering if this variant, if I could find evidence that this variant is pathogenic. So in the varvis suit, you have here the outlink to Mastermind, which is quite nice. And by consulting Mastermind, I found that I have almost 60 articles about this variant. So I don't want to read all of them. I need the information, but I need it in a reasonable time. And what I like very much about Mastermind is that you can ask the right questions. So Mastermind is not only a literature tool, but it's sort of an intelligent search tool. And the questions I had in mind, is there a report or a paper reporting that this variant has been found in a compound heterozygous state with another pathogenic variant? Then I could invoke PM3 here. Or are there functional studies? This would be criterion PS3. Or is the prevalence in affected increased over controlled? So is there a case control study? Then I could also use PS4. And how I did it is so this is the variant, I check the variant, then you go navigate to filter categories, navigate to this ACMG interpretation window, and I clicked on functional data and functional studies here. And then I submit the data, and then the few changes, and here in this publication history I have a large circle, which is this first paper here. And when I click on this paper, then I get here, I hope you can see it, I activated here variants only, and I have the keywords and the variants in one row, and so this was a clear shortcut. So again, remembering my first rule, this is not the evidence you need to, now we have to look for the evidence in the paper, but at least I have a shortcut, a serious shortcut to find the right paper to read. The same is true or can be used for the question if I found the variant in a compound heterozygous state. I navigated to inheritance pattern, zygosity, compound heterozygosity, submit with the other filter set. And again, voila, I find here that it was compound heterozygous with a stop variant and so on. And last but not least, I can look for case control studies here by choosing this setting. And I find the quote that this variant is really has a high prevalence, so almost 6.6% of all pathogenic variants is the specific variant. And yeah, this is my last slide, and I'm happy to answer. Sorry for the rather quick run through, but I want to keep in time a little bit. And yeah, you can ask later questions if you want. Thank you.
Okay, thank you, Andreas, for your very interesting talk. Nice, nice examples. I do see some questions coming in, but we will keep them for the Q&A later. So this will take me to the second speaker. There we are. So as our second speaker, I'm happy to introduce to you Dr. Ben Liesfeld, who is a physicist by training and has many years of experience in the development of medical devices, especially software as a medical device. He's the co-founder of Limbus Medical Technologies and also the managing director. And today, he will talk about how to master the flood of new data. So Ben.
Thank you, Yvonne, for the introduction. Also trying to get control. Okay, so Andreas Lahner nicely explained what the challenge is in clinical diagnostics to identify the clinical evidence that you need to support a specific classification of a variant. I would like to put this into a larger perspective, to the perspective of a clinical laboratory who needs to perform diagnostics on hundreds, even thousands of patients per year. So where a lot of data is pouring in just in form of patients, and that has also to deal with the vast amount of clinical evidence that is around there that is constantly changing, and how such a clinical laboratory can master this flood. I would like to give you some numbers from our point of view, how big this flood actually is. In some areas, genomic information is growing exponentially. Just as an example, the number of patients that have been analyzed with varvis in the past 2 years has been growing by more than 50% year over year. If you look at just the time range between November last year and November 2020, For example, ClinVar has grown by more than 50%. Almost 300,000 new unique variants have been added to ClinVar in that time frame. And another important number is the amount of raw sequencing data that our customers archive on our software platform. The amount of data has grown by almost 100% in that time frame, which means, or which illustrates nicely, that most clinical laboratories have been moving from targeted panels, small targeted panels, towards whole exome sequencing. So if you think of these challenges that you're facing in the laboratory, I would like to explain 3 important principles that you should apply in your workflow and your diagnostic processes that will help you to tackle this flood of data. The first principle is capture the data in a structured way. This is extremely important and extremely basic. If you think of the individual patient that you're diagnosing, don't just remember the single variant that you may have reported or may not even have reported. The entire genotype of the patient is actually very important. The entire genotype contains information that you can utilize to support classifications of other variants in other patients that you're seeing. In fact, you would like to create a structured database of all the genotypes that you've ever seen and create a database and put them into a database that gives you transparency in how many patients have you seen a variant and what are the phenotypes that these patients have been associated with. So this transparency gives you power, gives you knowledge, and helps you to assess clinical cases downstream. Varvis only supports kind of the right half of the NGS workflow, the bioinformatics, the filtering, classification, and the reporting, but it allows you to capture information from the entire NGS process. So, for example, any kind of information that is related to the quality of the data. And it also allows you to capture family relations and clinical information related to your patients. In particular, the clinical information, the phenotype, is important and extremely helpful solving genomic cases. Varvis tries to help you as much as possible and facilitates the capture of phenotypic data, so you can navigate through the ontology, the human phenotype ontology, and select exactly those specific terms that are relevant for your patient. And you can also store family relations in the database. That means that if you, in a later moment in time, add another family member to your database, you will be able to view this entire family at a glance together, and you may be able to extract a clearer picture, and you may be able to create a diagnosis with this additional information. And as Andreas Lana pointed out, it is extremely important that you and your team and your diagnostic team comply with standards and work in the same way, in a systematic and a consistent way with variant classification. So we support the use of the ACMG guidelines and provide you with a calculator that nicely allows you to document how you came up with a specific classification. So the criteria that you enter in the software tool are stored with the variant, are stored in the database, and are available for any future review or for any other patient that you are going to see this classification in. The second important principle that I would like to draw your attention to is that you need to be able to deal with the changing nature of the information related to genomics. Almost everything that we look at, all the information that we consume in genomics, is undergoing change, maybe except for the genotype of the patient if we are talking about hereditary diseases. But everything else is changing. For example, Varvis provides you with annotations for a variant from a wide range of sources, RefSeq, GNOMAD database, but for example also ClinVar. There may be, ClinVar releases new information every month, and there may be 100,000 new variants in such a release. Your own variant database in varvis is growing every day. So every day you will have new information that you can utilize and that you need to utilize to diagnose your patients. How can you deal with that? You need to be prepared to deal with the changing nature of that information. Well, first of all, varvis allows you to very easily update information. If you open any case in from any point in time in your analysis history, you can click on an update button and you're presented with information. What was the status of the information that was used back then when you performed the analysis, maybe 2 years ago, and it also presents you with information. What is the current information that could be applied now? And with the press of a button, you can update this information. But now keep in mind that recommendations from ACMG and also from the European Society of Human Genetics make the laboratories, the diagnostic institution, responsible for following variant classifications in public databases. So there's a certain level of responsibility that if such a public classification changes, that you are actually required to report this back to your patient if it is relevant to him. But how should you do that if there are bulk changes of 300,000 variants at a time? varvis provides you with a tool for that. So we boil down all these changes, all this new information, and only show you the information that is relevant to your patients. And by filtering, for example, regarding the criteria, what has changed and which patients have been affected, you can nicely navigate exactly to those patients that need review. So in this way, we intend to enable you to cope with this, with the flood of data and with the immense change that is happening every day. The third principle that I'd like to explain is, or that I would recommend you to do, is aggregate your data and leverage the knowledge that you gain by aggregating data. A single data point is almost useless, but viewing many data points together can give you an enhanced insight. And as I explained, varvis allows you to capture a single patient in a more complete way regarding all the variants that you find, regarding the clinical information that you have about that patient. You can then build up your own local database of variants of patients. You have full access to this analysis history, and it's typically a rather homogeneous data set. And on top of that, we create an aggregated database across all of our customers, which allows you to compare your data sets to other institutions, which provides you a lot of quality control information, but which also informs you about reported variants from other institutions, and of course also with information about the phenotypes that are associated with those patients. I'd like to give you some facts and figures what is provided with that database. At the moment, there are more than 24 million unique variants reported in this database. The data stems from more than 48,000 patients distributed across 25,000 families, and more than half of those patients have been described with standardized phenotype descriptions, which is a significant fraction and which helps you to judge and to interpret the data in more detail. Please note that we are also collecting information about copy number variations because our software also generates information about copy numbers. And this has started a couple of years ago, and we've already created data sets from about 16,000 cases in the database. If you'd like to learn more and read more about those aspects regarding clinical diagnostics, I would recommend that you check out our blog. There are a number of interesting articles in this blog that refer to the challenges in clinical diagnostics, and feel free to contact us if you have any questions. Thank you. Yvonne?
Yes, thank you, Ben, for presenting your data. I'm continuing with the third talk, so I would like to introduce to you Dr. Mark Heel. There you are. Hello, Mark.
Hello, Yvonne.
Mark completed his MD PhD in molecular genetic pathology fellowship at the University of Michigan. His research focus was on stem cell biology, genomic profiling, and clinical bioinformatics. Since 2014, he's co-founder and chief scientific officer of Genomic. And today, he will tell us about comprehensive literature search and how Mastermind is the entire world of genomic evidence at your fingertips. I'm handing over to you, Mark.
Oh, thank you so much. And thank you to Ben and Andreas for that excellent overview. I plan on emphasizing some of the points that were made, particularly when I describe why Mastermind is the most comprehensive source for genetic and genomic information built on the groundswell of evidence that's increasingly being published in the medical literature. So Yvonne, I'm just waiting to get control of the slides there, but if you have control, if you could advance there, that's great. So Andreas brought up the idea that one of the most challenging places to need to extract evidence from is the empirical evidence published in the medical literature. That has been a focus of Genomenon's in producing the Mastermind genomic database, and we are the gold standard, the most comprehensive source of such information, comprising as it does the 30 million titles and abstracts, where increasingly year over year, more than 500,000 new genomic articles are published. Mastermind is capturing evidence from each of those. To date, we have nearly 8 million such articles in our database, including content from 2 million supplemental data sets. With the increased use of next generation sequencing and the larger scale clinical and research studies being published, increasingly that evidence is being put into the supplemental data. So Mastermind is particularly good at capturing that evidence. And what we extract from that evidence is the mention of any disease in the whole spectrum of human disease, any of the tens of thousands of genes in the human genome, and all the different ways that they can be described, and any genetic variant that's described in any of those references. Those are all captured and indexed and kept up to date in the Mastermind genomic search engine. But Mastermind isn't just a repository of information, as Andreas alluded to. It is an intelligent system of genomic associations. So I'd already spoken to on the previous slide how Mastermind is organizing information around genes and their variants and how they relate to disease causation. That was articulated in its utility by Andreas in his use case examples. We also capture information around how those diseases, genes, and variants associate with therapies or with clinical phenotypes. In addition to, and particularly useful when interpreting this information according to ACMG-AMP guidelines, how they're interacting with information that's relevant to clinical determinations as well as functional information, such as in vitro or in vivo studies. And when I say that this information forms a sort of intelligence in our database, that Andreas spoke to how you're able to extract, and Ben highlighted how the integration of Mastermind into varvis through the partnership with Genomenon and Limbus deepens your awareness of the value of this information and streamlines and improves the accuracy of your variant interpretation process to promote more accurate, more complete diagnoses, including divining treatment therapies for your patients. And that's exemplified on the right, where the strength of these associations is built on the groundswell of that evidence that Mastermind is collecting and aggregating. So when you put this all together, as I said, I'd like to emphasize some points that Andreas made that I thought were really apt. He said, when referring to Fight Club, that the first rule is to verify, to gather and assess the evidence yourself. And Mastermind, as was shown, makes that process very straightforward and as complete as is possible. It's also useful in resolving conflicts. I think that there was mention of some of those conflicting points of evidence when Andreas talked about Karl Popper and the idea that your goal as a variant interpreter is to organize all of the evidence you can and assess it critically to refute your hypothesis. And if you fail to, then that becomes your interpretation. So Mastermind is particularly good at helping variant interpretation have all of the data and all of the evidence assembled at their fingertips to resolve those conflicts. And the third thing that I'd want to emphasize is when there are disease and or gene-specific interpretations that are required. Mastermind, particularly through its intelligent genomic association capability, is very helpful for the user to streamline their search and to either identify these associations de novo or otherwise to ask and answer specific questions that the interpreter is searching for information for. Andreas did an excellent job walking through the use example, so I'm going to spend just a few moments talking to some of the features in Mastermind. This first feature is just to underscore that Mastermind is a very exhaustive data source. There are multiple ways that you can describe a gene or a variant, multiple ways that you can refer to a phenotype or a disease or a drug. Mastermind, with its intelligent indexing capability, is aware of all of these ambiguities, different varieties, previous names, acronyms, etc. Particularly critically important when you're talking about the challenge of recognizing genetic and genomic information with all of the different ways that an author can describe such entities, including challenges with things like different transcripts and different legacy nomenclatures, the fact that we index for RSIDs in addition to protein levels changes and cDNA changes, and then allow the user to search on those different nomenclatures as well. So a simple query affords the most sensitive result set when you execute on that query. Here's what the Mastermind interface looks like in its fullest embodiment. Just to highlight those features, on the right is the literature landscape with all of the references citing that variant, prioritized according to clinical relevance, that is modifiable with user inputs from keywords or diseases, phenotypes, or drugs. We show our work. We show you a sentence fragment sufficient for you to confirm that this is indeed a reference that you're interested in reviewing. And then we invite you to review that information in full through the publisher's site. And then on the left, we have the full variant landscape to allow you to put your interpretation for that single variant in context. In this case, I'm showing the single genes variant landscape, but as I emphasized before, you're at liberty to add multiple genes, multiple variants, and review the information in the fullest context of that patient case. So this is easy search and easy display of information, affording you the maximally sensitive view into the evidence. Also touched upon that I'll just quickly re-emphasize is Mastermind's ability to maximize the specificity of your search result, as Andreas walked through in specific use cases. Ben, toward the end of his conversation, talked to the value that's provided through varvis by allowing users to search for and capture copy number variants, an increasingly recognized important component of genetic diagnoses. Mastermind, within the past several months, has released copy number variants capability for our users to search on copy number variants. As you can see here in my example, not just for constitutional disease or ACMG, but also for somatic disease and cancer, which is helpful when you're interpreting variants according to the AMP guidelines. And just as I had suggested before for Mastermind's intelligent ability to deal with the ambiguities and irregularities of genetic and genomic nomenclature, that is particularly true of copy number variants, where there's less widely used, less robust codification of an appropriate nomenclature that's agreed upon for describing copy number variants. Mastermind recognizes all of the different ways that copy number variants can be described. puts those together to maximize the sensitivity of your search result, and then allows you to review that information and refine your search with enhanced specificity search capability. So in this case, you'll see in the upper left our ability to find all of the different EGFR deletions, whether they comprise commonly deleted exons like exon 19 or 20, or whether they're larger or they're less frequently mentioned exon deletions, such as may be present in your workflow in that particular case. So the last thing I want to emphasize is that all of the attendees are invited to sign up for Mastermind and be afforded access to Mastermind Professional, which we have talked through in Andreas's talk, and which Ben had spoken about with respect to the Limbus integration. Sign up with that bit.ly link, and you'll be afforded the most sensitive, most robust genetic information to pattern your variant interpretation. So with that, apologies for running over a little bit, but I'll pass it over to Yvonne to field any questions that have arisen.
Great. Thank you so much also to you, Mark. And for the last part of our session, this Q&A session, I would like to welcome all speakers back here. And indeed, there are quite a few questions that have been entered into the in the chat window. I hope we can get through all of them. The first one is for Andreas. Would you review a ClinVar entry with a 4-star rating to the same degree as one with a 1-star rating? You are mute again.
That doesn't work. Well, so this refers to my first rule in variant interpretation, or to Fight Club. So of course, this discussion comes up very often, and of course there's a difference. But even a ClinVar approved 3-star or 4, so the 4 stars are not questioned. So I don't question these anymore. With the 3-star submitters, there is so I'm myself a submitter of a 3-star genes in ClinVar, and of course we are humans and we make mistakes. And for some of these even 3-star curated variants, we had to change our classifications. But so one part of the answer is there is no reputable source, and the other part is even the VSEPs from ClinGen, they show their rules and they show their evidence codes, so you can follow why they come to a class 4, 5, or whatever. So if they calculate a probability of more than 99.99%, then it's quite unusual that this class 5 will ever be downgraded. But still, it is a possibility, and you can follow it.
Thank you. There's a second question for Andreas. Based on your experience, have you reclassified variants already reported to your patients? And if yes, how often?
Well, not that often, but yeah, that's the real nature of variant classification. So a classification of variant is almost never static. So as we heard from Ben and also from Mark, the field changes so incredibly fast in the last years, and I don't think that this will stop. So just in the beginning, when I started classifying variants, we were doing Sanger sequencing, 5 patients per week, and we were quite happy when we found a missense variant. We did this HPLC stuff, and it was quite weird. And many of the variants we thought that they were pathogenic were indeed benign polymorphisms, but this was evident only after GNOMED and EXTEC and ESP and 1000 Genomes published their data. So and we are never finished here. So of course, reclassifications occur and newer data sets. So there are some good data out from the Young et al. Paper from Invite, from these guys, and from the ClinGen consortium. They show that newer classifications up from 2016, 17, 18 are better than older ones. Of course, this is true because there are many new data came in, but still in the year 2020, we have to reclassify variants from time to time.
Do you support classification of variants in association with a disease of interest? For example, a BRCA2 variant with one class in breast cancer, and with another class in Fanconi anemia, meaning multiple classes per variant.
So ACMG says one classification for one variant for one phenotype. But in the case of BRCA2 and Fanconi anemia, I would say that in both cases, loss of function variants are causative. That's only the mode of inheritance is different. And I would question if there are for one clear-cut pathogenic Fanconi anemia, for two variants in Fanconi anemia or homozygous state, then this should be also causative for breast cancer. But there are many other genes where we have many more phenotypes like LMNA, or there are many examples, and there you must state in the report for which variant and which phenotype you come to this in this classification.
Okay, the next question is for Ben. How often are annotation sources updated in varvis?
Well, generally this depends on this annotation source, of course. For example, ClinVar releases new data every month, and that's the interval at which data is updated. The ClinVar data is updated in varvis. So far, we have not had sources that update more frequently, but we would try to keep up with the evidence as it appears and as it is released.
Follow-up question for Ben. Does the user get an alert if the classification has changed, or are the alert tools only available if you actively search for them?
Well, so far in our experience, I mean, depending on the number of patients that you have in your database, there may be a larger number of alerts. So we found it most useful to view them together in a specific view in the software and not alert the user one by one. So it's a comprehensive view of all the alerts that are generated.
Okay.
If you don't mind me jumping in, Ivan, I think I mentioned it earlier, but just to state that Mastermind is updated on a weekly basis, and we have an API that allows you to access all of the most up-to-date information, and we make that information available through our integrations as well. And then also to say that I think there's increasing importance around looking back at your previous interpretations. So that was a really good question about look backs and the benefit of being alerted to any new information that might change your diagnosis. So that's one of the benefits of staying up to date with the information, and Mastermind through our platform allows for users to subscribe to Mastermind alerts to be continually updated to that new information.
Yeah, there are quite a few questions regarding Mastermind, and one question that is asked frequently here is Mastermind a free online tool?
It's a great question, and that slide that I presented with the link will give you access to the paid-for professional version for some 3 days to allow the user to see the benefits of the features that Andreas had spoke to, some of which are integrated in the varvis system. But there is a basic edition that has limits on search capability that is available to the research and academic community, although the professional version is highly recommended for clinical workflows. But yes, indeed, there is a Mastermind Basic free edition.
There's also a question regarding the professional edition. How much does it cost, and how many people can work with it?
I'll defer questions on cost to my sales team so I don't get into trouble. But we're interested in making this data as widely available as we can to promote increased diagnostic yield and accuracy, and to make this evidence available to the benefit of patient care. And so if you're talking about institutional access or access through some integrations such as Genomic has with Limbus into the varvis platform, let's have a conversation about how many users are at your institution, because as I said, we do want to get this information into the hands of the clinicians for the betterment of the patient care. I hope that I answered that diplomatically enough.
How is the Mastermind data on a variant collected, and more importantly, verified?
Great question. So we've spent the better part of 8 years with a large development team, so many man years, in refining our ability to find this information. So the first is where is this information? And I spoke to our content, particularly in terms of our ability to go at the full text level, figures, tables, and supplemental data. Our philosophy is sensitivity first and specificity later. So in our indexing capability, we recognize all of the different ways that a variant can be described, and we've patterned that into our indexing technology, including, as I mentioned, the transcript differences and the legacy nomenclature. So the goal behind our indexing is to be certain that we're not missing anything. Different gene names is a big one. If the gene is described in a certain way in a paper, we want to make sure that we find that by having the most sensitive search. Once we've aggregated all that information, it's left to the user, as Andreas had shown, to verify that that's correct information. And the profile is quite good in terms of sensitivity and specificity, in the high 90s for both. But we show our work. We present you with the evidence and allow you to verify. Trust but verify is, I mean, overall in variant interpretation is a great mantra. We believe that too when you're looking at the evidence that we've presented in Mastermind. And then I'll separately say that for many of our larger diagnostic labs, as well as our pharma partnerships, we curate this information internally. So all of the work that Andreas spoke to, we're very well aware of at Genomenon, and we do so on a large scale, whole genes at a time for thousands of variants, including whole gene pathways can be curated and annotated according to ACMG-AMP criteria as well.
Okay, and we have another question for Andreas. How do you deal with risk factors variants? Will they be incorporated to the ACMG criteria system as well, do you think?
Well, explicitly risk variants or pharmacogenomic variants and multifactorial variants are not covered by the ACMGs, but of course, some of the logics apply to them. So you can use the ACMGs to classify them, but you have to keep in mind that some of the rules will make nonsense. So homozygous state in highly penetrant phenotypes, where we have the wording of phenotypes, penetrance, and so on, you cannot use it. But of course, some of the criteria can be used. And we do not regularly report. So I'm a little bit biased because I'm a hardcore Mendelian genetics, rare disease, maybe in the cancer type, but not pharmacogenomics and risk factor GWAS things. That's another thing. We do that as well, but we report this with another software with polygenic risk score calculation. So this is maybe there are some words now becoming overlap, but right now I like it separate.
Follow-up question: what is your opinion on the use of AI ACMG pre-selection tools?
Was this question for me or?
Yes,
maybe
also for you.
Yes, I work. I don't work with them. I try them. I try a lot of them. This is part of my job at MGZ to be up to date. What's going on? Tried several of them, and but still, maybe that's a bias because I'm so long in business. I like the hard data and the hard things, and I like to understand why a variant is pushed up or down and prioritized. But I'm well aware that in a couple of years we will routinely use these tools. And I like to understand from the very beginning where this goes. And yeah, so I have some good examples. I had some cases solved actually by AI tools, and but most of the time it was that this, yeah, it is at this time a little bit disappointing. But I'm sure that this will be a hot topic in the near future.
I'd like to re-emphasize what Andreas said at the beginning: is trust no one, or trust but verify, and that includes AI systems, where Andreas says there's a great need to see the hard data. This is true across all AI. It's no longer acceptable to just have it be a black box. We need to know the provenance of that evidence and how that the interpretation came to be, especially when we're talking about diagnostic care. And so, as I say, Genomenon in Mastermind puts a great deal of primacy on showing our work and directing users to review that evidence on their own, even despite our use of AI capability. So I would agree with Andreas. in getting the work done yourself, including when that work is resulting from AI technique.
I totally second that. Limbus, we are ourselves conducting research with the University of Rostock, in particular with respect to AI and being transparent. I mean, using AI in a medical device, just the regulatory process needs to be transparent, and the decisions that an AI makes needs to be transparent. And provenance is a very important keyword there.
Right, right.
Okay, last question. So I think we're running out of time. How do you implement the latest suggestions for variant classification? Especially I ask regarding the recommendation to downgrade PM2 to PM2 supporting. Who wants to take this question? It's not stated to who this question is. Ben, do you want to comment on that?
Well, we are continuously updating, of course, the software. ACMG guidelines are changing, and this is probably also true for Mastermind. I mean, we have to keep up with improvements in the guidelines, and as we try to do that as quickly as they are published to incorporate this into source code, and we make transparent what the version is and what guidelines we are following, what compliance you can expect in the software.
Okay, there's also a question that we can get the presentation PowerPoint files, but I think we will distribute the recording of this webinar so everybody can take a look and recap. And yes, I think I would like to conclude. I thank you, speakers, all of you three, for presenting the nice data and examples. For more information, please visit varvis.com or contact info@varvis.com. Also, if you have still questions in mind, we are happy to forward them to our speakers. We will have some more sessions of the varvis webinar series coming up next year, so make sure you sign up for our newsletter to don't miss future sessions. And by this, I would like to say goodbye to you all, and good night and good evening, and happy Christmas.
Thank you.
Bye-bye.
Bye-bye.
Thank you.